Alors, RhizoSeq-C en français n'a pas d'homophone. But if we switch to English, then it becomes RhizoSeq-C, and it could be a little bit confusing. So I do confirm that we are dealing with rhizodeposition and carbon sequestration in the project. So RhizoSeq-C, no, sorry, I will try to move with my computer. So I apologize, I need my computer to, I'm not very good English as Jean was. So in conventional agricultural systems, most soils continue to lose carbon, and this trend is represented by the gray curve. But it can be reversed through the implementation of agroecological practices, as shown by the green curve. And the mean range of additional storage is estimated to be 0.3 to 0.4 around ton of carbon per hectare per year. And all of these practices are agronomic techniques that do not take into account the intrinsic potential of cultivated species. But however, the intraspecific variability within these species could also represent a lever that we propose to explore. So it's now well established that rhizodeposition of organics through root exudation, mucilage deposition, or degeneration of root cells represents a major input of organic matter that feeds into the stable soil carbon pool by associating with mineral phases. So our consortium thinks that genotype selections toward increased rhizodeposition may enhance root carbon inputs to the soil and could therefore be a promising, easy-to-implement management option for potentially increasing carbon sequestration. So the quantification of rhizodeposition is, however, still quite a challenge, and the most accurate methods involving isotopic techniques but they are difficult to employ for large-scale phenotyping. However, rhizodeposits are strong binding agents, and the rhizoschist represented in yellow on the diagram, and I'm not sure that you can see the yellow, samples the root adhering soil, and this is therefore considered as a good proxy for rhizodeposition. And the example of phenotyping obtained on pearl millet shows that the rhizoschist size varies by a factor of 3, but also that the size of the rhizoschist is genetically regulated by multiple genes. But if we want to understand the mechanisms that sequester carbon in the soil, rhizodeposition is not the only factor to consider. This diagram illustrates the inputs of organic compounds into the soil, either directly through the rhizodeposits in red, or via microorganisms that biotransform a portion of them in blue. It is shown that genotypes with different rhizodeposition traits also correlate with changes in rhizosphere microbiota structure and functions. It's also necessary to consider changes in the rate of mineralization of organic matter already present in the soils, known as the priming effect. As well, if we want to focus on carbon sequestration in the soil, it's also crucial to consider soil types and their mineralogy, which determine the nature of organomineral interactions that stabilize organic matter. And finally, all of these processes are directly dependent on climatic conditions, which must also be considered as variable. Thus, carbon sequestration is the result of interactions that involve not only carbon inputs into the soils, but also its fate in the soils. And if we want to optimize soil carbon sequestration, all of these interactions must be explored. So the overall objective of the project is to develop cropping systems capable of increasing direct soil carbon inputs and promoting soil organic matter stabilization while maintaining economically attractive production in diverse climates. The project is thus divided into 6 interdependent sub-objectives. The first one, at the plant level, is to identify the genotypes and genomic regions that promote carbon inputs via rhizodeposition. At the agrosystem level, the second objective is to understand the mechanisms and determinants of carbon transfer within the plant microbiota soil continuum. The third is to identify minerals that promote organic matter stabilization by organomineral interactions. The fourth is to estimate the feasibility of implementing such cropping systems while gently considering the agronomic benefits, the extent of water stress, and climate change. And at the impact level, the objective is to quantify additional soil carbon storage to assess the sequestration trends in the long term. The last objective is to organize a carbon sequestration community of multidisciplinary scientists. Regarding experimental choices now, our focus is on sorghum. Sorghum ranks as the 5th most cultivated cereal worldwide, characterized by high biomass production and tolerance to water deficit. Additionally, back-cross-nested association mapping lines are available for sorghum. And sorghum is also closely related to pearl millet that we have seen just before, for which results on RhizoSeq genotypes were very convincing. Concerning the choice of soils, we have chosen arenasols for several reasons. As depicted in the graph, arenasols constitute the 5th major soil type globally, particularly prevalent in African region facing high food pressure, yet they are relatively understudied. They typically have low carbon content and their carbon stocks are highly susceptible to land use impacts. Moreover, arenasols are characterized as poorly aggregating soils. Therefore, for this project, they provide a relevant substrate for easily demonstrating differences in aggregation and carbon concentration properties, similar to what has been observed for pearl millet. We will have also project tasks where we will test 4 other soil types that are particularly important in terms of surface area in metropolitan France, which are brunisol, calcosol, luvisols, and fluvisols. The project is structured in 5 scientific work packages and 1 coordination work package. It operates at 3 levels: plant level in green, agrosystem level in brown, and impact level in blue. It covers scales ranging from nanometer at the gene level to hectares at the cultivated parcel level. And it also spans time scales from daily for the priming effect to a quarter of century with modeling. And this project also has a strong multidisciplinary component. So the first work package focuses on plant and rhizodeposits, and it comprises 4 tasks. The first task involved determining the genetic diversity for rhizosheet formation in sorghum. The next step involves developing genetic approaches. We will also quantify total rhizodeposition on selected genotypes and characterize root exudate composition. Concerning the second work package, at the crop system level, rhizodeposits act as a recruitment signal for rhizosphere microbiota. Therefore, work package 2 will focus on the modification of the microbial compartment for contrasted lines, and it comprises 3 tasks. The first task will involve characterizing the impact of biodiversity of microbial communities. Next, we will focus on gene expression patterns of microorganisms, and then we will quantify the priming effect in relation to the nitrogen cycle. Then in the third work package, we will be interested in the influence of soil type, mineralogy, and climate on the fate of organic matter entering the soil through rhizodeposition. This work package comprises 3 tasks. The first will seek to measure the variability of net rhizodeposition as a function of soil type. We also aim to introduce highly reactive minerals into the soil in an effort to promote the formation of organomineral interaction to stabilize carbon. And we will seek to develop innovative approaches for monitoring the fate of carbon in situ in the soil using cutting-edge imaging tools. The fourth work package will evaluate the impact of changes in rhizodeposition on the agronomic performance of sorghum under field conditions. We will conduct experiments in microplots in France and in Senegal, focusing on the characterization of agronomic traits. And in parallel to work under realistic conditions, we will conduct trials in farmers' fields in Senegal for 3 years during the rainy season. Finally, the last work package will allow us to quantify the impact of these practices on soil carbon sequestration, and the work will be divided in 2 tasks. The first task aims to quantify the carbon sequestration balance over 1 cropping cycle in the field using isotopic balance approaches that couple 30C and 14C carbon. And the final task will predict the soil carbon sequestration induced by sorghum cultivation by 2050 using the AMG model that we have seen this morning. As a whole, the project includes laboratory experiments and field experiments conducted in 2 contrasted climates. And concerning the partnership, it consists of 10 research teams, and most teams are involved in several work packages, so that this will facilitate the exchange between teams. And then I will finish with 2 highlights. The first one is the project started on the 1st of October, and the kickoff meeting was organized 10 days ago, so it's rather fresh. And at the first RhizoSeq-C results obtained in October already show significant differences in rhizosheet formation for 14 sorghum lines. Thank you very much for your attention.
Thank you very much. I think we have a minute or so if anybody wants to ask for any specifics or questions. We have 2 minutes if you like. If you would like to ask for some clearer specifics. Do you want to sing us a song or something? Or Pierre has a question.
Thanks for the presentation. I was wondering, are there any relationships between the exudation and root exploration? Is it a trade-off, or on the contrary, if we have more exudation, do we have more root exploration, or is it just not related? Do we have information on that?
Laurent, could you help me, please?
Laurent, hold on. This won't be deducted from your time. Laurent, tell us.
As well, so there is no clear correlation between both. In fact, root exploration is linked to root elongation, and elongation is linked to rhizodeposition. It's can be a compulsory affair. So it's not a direct positive or negative correlation between them. And so in former panels for which we screened many lines, we find all the figures. So it's not so closely linked, but. Yes, maybe we will explore there more in detail in this program.
Thank you very much indeed, Laurent. Okay, well, I think that's, j'oublie votre nom, Isabelle? Isabelle, thank you very much indeed for that very well presented RhizoSeq-C project. Thank you very much indeed.